Okay, so welcome to the first of two components of your aerobic metabolic pathways that we'll be learning. And the first one is the Krebs cycle. But to that point, before we get too far into it, I want to at least introduce both of the pathways that are present in the aerobic metabolic system as a whole. There's two main biochemical pathways. The first one being Krebs, which we already stated. This will be the focus of this video. But the next video will focus on the electron transport chain. And that's the second part of the aerobic metabolic system. So just to, again, introduce a few concepts about this. Krebs cycle oxidizes substrate. So we'll talk about what a redox reaction is chemically. And what essentially goes on is this kind of removal or moving of hydrogen atoms and transferring of electrons between different chemical compounds throughout the Krebs cycle. It'll produce some, and by some I mean one ATP in the cycle, but it'll allow us to use derivatives from protein and fat, and essentially creates a bunch of byproducts with these hydrogens and electrons to be able to later be used by the electron transport chain, which is where we have the primary or the majority of ATP produced in our body. Again, as I mentioned previously in one of the videos, it's the most efficient means of producing ATP that our body has. So yeah, that's kind of a main focus if you want to know where the majority of your ATP comes from or comes for your body. Okay. The process is also known as oxidative phosphorylation, and that explains more as to what's happening in the actual processes. Again, we'll talk about what a redox reaction is. You don't need to remember the specifics of it, but it's good to see and understand as a general process. And ultimately, as I've already mentioned just a couple of times here, our NAD plus, if you remember from glycolysis, as well as a new Uber or taxi cab for hydrogens, FAD or flavin adenine dinucleotide, FAD. They're like the taxi cabs that you get your hydrogens loaded up on, and you can shuttle it to places that you need to have them go. So again, Krebs will produce quite a few of these. We get two from at least from our glycolytic system, and that will get shuttled over to the electron transport chain. Okay. Ultimately, it's aerobic respiration, so we need oxygen, right, to help us complete the cycle and produce ATP through this form of metabolism. And sure enough, it comes in at the end of the ATC. And what happens is water is formed when we get one oxygen that accepts a couple of hydrogens as a final step. And yeah, that's kind of where oxygen comes into play for the aerobic system, is at the end of this quite extensive biochemical pathway to produce energy, especially if you're thinking about the original step before glycolysis, where we have glucose starting its journey there, and if we follow the carbons on that structure. But I digress. Let's get a little bit of an overview here of what exactly a redox reaction looks like, what it is. Again, it's more of the generals. If you've taken chemistry, you probably already have an idea, or just need a little bit of a cueing of memory. But if we look at this very general process of metabolism, right? We have our glucose molecule here, our oxygens here, and what happens is as we go through metabolism, again, this is just a very general overview or way to look at it. What will happen is oxygen becomes or goes through a reduction, which means it'll gain some electrons. And through that process, that's kind of gained through hydrogen, right? So hydrogen, again, will attach to water. The chemical a formula or makeup, the atomic makeup of water is H2O. So two hydrogens, one oxygen. So that will split, and you end up getting a water. Now the other oxygen, obviously, we can find in carbon dioxide. And what will happen to our glucose, again, this is the or the red, or the reduction. The ox of a redox is the oxidation. So what will happen here is as glucose becomes oxidized, it'll lose electrons, and therefore ends up becoming carbon dioxide. Okay, that's the basis of that. Again, some people will be more familiar with this than not. The idea is just when we talk about oxidation, when we talk about as a process of metabolism, it's good to know exactly, or have a better idea of what's going on when we say that, what we mean by that. Okay, enough of direct chemistry. We'll dive back into the Krebs cycle, and we'll have a little overview of the whole process. But one of the first things that need to happen, and we spoke about this in brief, and we foreshadowed it when we were talking about the end products of anaerobic metabolism, or glycolysis more specifically, is we end up with a end product, or this end product called pyruvate. So that will be from glycolysis, and again, in the absence of oxygen, it'll end up being turned into lactate, if you recall. But here we're going to assume that there is oxygen present enough to continue on into aerobic respiration. So what will happen is pyruvate will get converted into something called Acetyl Coa. And just to help you work with this in your mind so you can picture what it might look like, is you can just picture an acetyl, the word, as like just a group of atoms that make it an acetyl group, and CoA is just a separate entity. So when they hyphenate this word, do almost picture that as like a chemical bond between the two, okay? All right, so acetyl CoA can enter the Krebs cycle, which is important because the Krebs cycle is where we go through oxidative metabolism. So it enters as a 2-carbon molecule, and one of the first compounds within this cycle is called oxaloacetate. So that's essentially like a 4-carbon long molecule, and the idea is throughout this entire circle, and if you haven't quite picked up on it yet, throughout all of metabolism, you're essentially following different lengths of chains of carbon structures, and they vary depending on what stage of what process you're at. So we get to this point now where we have our 2-carbon acetyl CoA attached to oxaloacetate. It's such a mouthful. That's 4 carbons, so we get this 6-carbon molecule in the end called citrate. Now, I bring that up because this whole process, and maybe you learned this in high school or at a different point in time in life, but this cycle was also called the citric acid cycle. And the main reason is we have this citrate molecule, the 6-carbon, that goes through the full cycle and ends up back as oxaloacetate, and because of that, again, we call it a cycle, right? So we used to have citrate as kind of this marker or initial molecule in the cycle. I digress. We'll call it Krebs. We'll keep calling it Krebs here. So citrate, as I mentioned, it goes through this whole process, and in the end, this is where we get our byproducts, such as, or some of our byproduct of carbon dioxide. We get NATP off of this, but one of the other key components is this is where we start shedding, let's say, a lot of our hydrogens and electrons. Okay, let's look at this. We'll look at the whole overview. I love visuals. This is such a good thing. You don't have to memorize all of these steps for this class. One day, chemistry, you might need to. But if we note, here's pyruvate at the start of the cycle. And so this will be the input from glycolysis. As a reminder, just following along to try to hit some of the main steps. We got pyruvate dehydrogenase as an enzyme working here right away. And the first thing that it kicks off, carbon dioxide. Okay, and through that we get a full Uber, an NAD, plus a hydrogen. Okay, so right away we're already producing some metabolic byproducts, which will be breathed out later, the carbon dioxide. The NADH, of course, that's our shuttle. That's going to bring things over to the electron transport chain in due time, okay? Coenzyme A, again, these are fairly common enzymes, so to recognize them maybe on a multiple choice would be a good idea. But we end up with Acetyl Coa, and again, if you recall, that's our starter molecule. That's the two combo molecule that we need to add those two carbons onto oxaloacetate and essentially create citrate, which is the first compound that we start using in the Krebs cycle. Okay, so that was our intro too. I used a bigger component here for the intro part of leading into the Krebs cycle. I don't know if the screen is large or small enough for you, but just so you can see pyruvate, CO2, and Acetyl Coa. Okay, so let's look at a bit of a blown up version of the Krebs cycle in and of itself after it has its precursors. The important things to note are what's coming out of this cycle. Okay, so we can see here that we have another NADH now that's full and ready for electron transport chain. We have another carbon dioxide that happens again and again, this time without the carbon dioxide. Our CoA molecule does end up coming in down here to the bottom of the Krebs cycle and helping essentially in a multi-step create our ATP. And we also finally get an FADH2. Okay, so this is our one more FADH2 shuttle, I guess you could call her Uber, that's going to take hydrogens over to the electron transport chain. Okay. So at the end of the day, what do we get out of the Krebs cycle? For each acetyl CoA that goes into the cycle, right? This is after glycolysis. 1 ATP, 2 carbon dioxides, 3 NADHs, and 1 FADH2, or 2 hydrogens. Okay. All right. Now, kind of just backtracking a little, going to summarize. Why do we want to keep track of all of this? Well, I know you're asking, you're like, hey, whoa, Sean, pump your brakes. You said the aerobic metabolism is going to give us the majority of our ATP that our bodies use. But I remember that glycolysis gives me at least 2, if not 3 ATP, depending on whether the initial source is glucose or glycogen. This Krebs cycle only gave 1. Where's all this other efficient ATP that's being produced? Well, you just sit tight. That's some foreshadowing for the next video where we talk about the electron transport chain. Okay, and you're going to see all of these molecules of hydrogen that we've just been cranking out, not producing ATP yet, is going to get put to good work. Okay, see you on the next video. Cheers.